Our next speaker is Franz Mittermaier from Charité in Berlin, and he's going to talk about synaptic connectivity in the human neocortical expansion.
So maybe I can start real quick by thanking Taufik and the rest of the committee for organizing this great meeting. I think we need some technical assistance. Here we go. Okay, so I'm very excited to present today our work on human neocortical layer 5. So this is all unpublished work, and some of the analyses are still preliminary. But in principle, I will discuss how this circuit scales from rodents to humans. So we've heard this a couple of times today, but when we look at the cortex of different mammalian species, then we observe that they differ vastly in size. For instance, the cortex of the human contains roughly 1,000 times as many neurons as that of the mouse, which is, of course, the commonly studied laboratory animal. Now, this raises a fundamental question, and that is, how do the circuits of the cortex scale to adapt to these vast differences in size? Of course, circuits of the cortex can be studied on various different levels, but in the Geiger group, we're particularly interested in the cortical microcircuit, so that is the synaptic connections between neighboring cortical neurons. When we talk about layer 5, we know from rodent work that the pyramidal neuron population of this layer can be separated into two broad groups based on long-range projection patterns. So we have extratelencephalic neurons, which project to subcerebral structures, such as the thalamus, brainstem, and spinal cord. And then we have intratelencephalic neurons, which mainly project to other cortical areas and the striatum. Now, these two groups do not just differ in terms of long-range projections, but they also show distinct morphology and electrophysiology, and there's also a number of marker genes that are associated with one group or the other. In addition to long-range projections, the axons of these neurons have early collaterals that give rise to a circuit within layer 5. And in rodents, a canonical circuit rule has been identified across different cortical areas that basically says that E.T. neurons connect only sparsely onto I.T. neurons, so that is to say with a low connection probability, whereas I.T. neurons connect densely onto E.T. neurons. Now, when we compare the central nervous system of the mouse to that of humans, then we observe that in the mouse, the subcerebral structures, so shown here in red, are quite large compared to a relatively small neocortex. In humans, the opposite is the case. Here we have an expanded neocortex that is quite large compared to subcerebral structures. In line with this, we know that E.T. neurons are more sparse in the human neocortex. So the idea here is that throughout human brain evolution, the I.T. neuron population has expanded drastically, whereas the E.T. neuron population and its associated subcerebral target structures have expanded to a lesser extent. And therefore, we end up with the change in cell type proportions. So focusing on the temporal cortex, in the mouse, for every E.T. neuron, we have approximately 4 I.T. neurons, whereas in humans, for every E.T. neuron, we have 20 I.T. neurons. So how is this relevant for the microcircuit? Well, every time when there is a change in cell type proportions, as a direct consequence, there has to be an associated change in the circuitry in one way or another. And to demonstrate this, I want to show two theoretical scaling scenarios that make this point clear. So we start with the mouse situation where we have 4 I.T. neurons for every E.T. neuron, and we have some connection probability between them. The first scaling scenario could look something like this. So we add I.T. neurons to get to the human situation, but we keep the number of synaptic connections that converge onto the local E.T. neuron constant. Therefore, I term this the stable number of connections scaling principle. In this case, the connection probability, so that is the chance of any I.T. neuron to connect onto the E.T. neuron, drastically decreases. An alternative or opposing scaling scenario could look like this. Again, we add I.T. neurons, but this time each added I.T. neuron connects onto the local E.T. neuron with the same connection probability as the ones that were already present. So therefore, the connection probability now stays stable. But as we can see, now the local ET neuron receives way more synaptic input connections. So with these theoretical considerations in mind, we set out to determine which of these scaling scenarios more appropriately describes human layer 5. To do so, we prepared acute brain slices from the middle temporal gyrus of patients undergoing neurosurgery. And I then performed multi-neuron patch clamp recordings in layer 5 of the human cortex. Now, as Yang Fang has already shown earlier today, what these recordings allow us to do is, for one, we can screen for synaptic connections between simultaneously recorded neurons, as shown here in this connectivity matrix. And from this, we can reconstruct the local circuitry. In addition, we can also characterize the intrinsic electrophysiological properties of each neuron that we record from, and we can reconstruct their morphology. And this is actually key to this project because we use these intrinsic properties to determine the projection type of these neurons. So how does this work? Well, traditionally, if you want to determine the projection type, you need to perform retrograde tracing experiments, right? You need to inject a tracer into some target region, and this will then label neurons that project to that target region. And this is obviously something that we can't do in human subjects for ethical reasons. Fortunately, there's an alternative approach, and here's how this works. So in 2019, the Allen Institute demonstrated that you can use single nucleus RNA sequencing to align cell types, transcriptomic cell types, across species. Using IT neurons as an example, here's how this works. So in the mouse, there are transcriptomic cell types where we know that their projection type is extratelencephalic. It is then possible to identify a human transcriptomic cell type that matches these mouse types. And from this, we can infer that the projection type of this human transcriptomic cell type is most likely also extratelencephalic. Now, in a subsequent study, Brian Kalmbach and colleagues used the patch-seq approach. So this gives you both the transcriptomic data modality as well as intrinsic electrophysiology and morphology. And what they found was that human transcriptomic IT and ET types differ enough in terms of their electrophysiology so that it becomes possible to predict the projection type based on electrophysiology alone. What we then basically did is we took the data from the Allen Institute to train a classifier and then predicted the projection type of the neurons that we recorded from using electrophysiology. Now, I don't have time to show all the differences in electrophysiology between human IT and ET neurons, but since seeing is believing, I want to show two key properties. So here on the left, you see traces of a typical IT and ET neuron in response to a chirp protocol and a subthreshold step protocol. And as you can see, human ET neurons are characterized by a resonance frequency in the 3 to 5 hertz range and a low input resistance. And if you just use these two properties alone and plot them on a scatter plot, you can see that this already distinguishes the two populations quite well. As a proof of concept, if you use electrophysiology to classify neurons and then study their morphology, then you will find that the cells that are classified as ET neurons have a large soma, thick apical dendrite, and profuse branching in layer 1, which is consistent with what we know for the cell type from rodent studies. Now, going back to the multi-neuron recordings, with this classification approach, we can now study the circuit with a cell type resolution. So the first thing that we did was we looked at connection probabilities between different projection types. And what we found was that human ET neurons project with a very low connection probability onto neighboring IT neurons. Conversely, human IT neurons project with a very high connection probability of more than 20% onto ET neurons. Now, this is pretty cool because it mirrors what we know from rodents. And we can basically conclude that the microcircuit principle of directional flow of information from IT neurons onto ET neurons is a principle that is conserved from rodents to humans. And this tells us that this is probably important for how layer 5 works. Now, another conclusion that we can draw from the connection probabilities is this. So, as I said, IT neurons connect with a high connection probability of 20% onto ET neurons. And this connection probability actually matches very closely what we know from rodents. And so going back to the scaling scenarios, this suggests that the human layer 5 scaled according to a stable connection probability scaling scenario, which means that human ET neurons have to receive more synaptic input connections. In fact, if we use the changes in cell type proportions and the connection probabilities that we obtained, then we can estimate that a human ET neuron receives 5-fold as many IT inputs compared to its rodent counterpart. Now, another way to look at this is by looking at the distance dependency of connectivity. So what you see here are all the IT neurons in our data set and their position in relation to the ET neuron. Gray dots are those that don't form a connection onto the ET neuron. Blue dots are those that form a connection onto the ET neuron. So what this plot shows is that neurons, IT neurons that are closer to the soma of an ET neuron connect with a higher connection probability, and then the connection probability drops off. However, in humans, the sigma of this Gaussian fit here is quite large, with 343 micrometers. We can then compare this to rodents. So here you see basically a very similar plot from the Campagnola et al. Paper. And you see that the distance dependency in rodents is much stronger, so the connection probability drops off way quicker. And you can then take the densities of IT neurons that we know from published literature, and you can use the distance dependencies and the cell densities to compute basically how many IT neurons are in reach of an ET neuron to form a connection across species. And when we do this, we find that a human ET neuron will receive inputs from approximately 2,000 IT partners, whereas a mouse ET neuron will receive inputs from approximately 460 IT neurons. So again, a factor of 4 to 5. Now, an increase in input connections is, of course, if you say 5-fold, a bold claim. So we said we want to use a different data modality to test whether this is even plausible. So we set out to study dendritic spines in ET neurons across species. To do so, we used the mouse SIM1 line, which labels ET neurons. And we limited our analysis to the basal and apical oblique branches, because this is where we would expect local synapses to form. The first observation that we made is that human ET neurons have, of course, longer dendrites, 2 to 3 times longer. However, the dendritic spine densities of the basal dendrites are almost identical across species. And for the apical oblique dendrites, the spine densities even slightly increase for humans. So when we use the total dendritic length and the spine density values, we can compute the total amount of dendritic spines per neuron. And when we do this, we find that human ET neurons have roughly 3 to 4 times as many dendritic spines as their rodent counterparts. So this gives us some confidence, and we can claim that at least from an anatomical perspective, it is plausible that human ET neurons receive more synaptic input connections. Now, this is kind of a tangent, but it builds on what Yangfang presented earlier today. So we also performed a second-order motive analysis to gain a deeper understanding of the circuitry. So in principle, there are 4 different second-order motives. We have the convergent, the divergent, the chain, and the reciprocal motive. However, when you consider that we have 2 different cell types, then this turns into 23 unique motives. What we then do is we count the appearance of each motive in our empirical data set. So we just count how often we observe a certain motive, and when we then compare it to a random connectivity model. And this can then tell us which of these motives are over or underrepresented. And there's a lot of information in this graph, but I want to draw your attention to just 2 motives. So what we find is that in particular, the convergent motive, where 2 IT neurons converge into an ET neuron, are significantly overrepresented, but also the divergent motive, where 1 IT neuron projects onto 2 ET neurons, is also overrepresented. So this basically just underscores the strong tendency of IT neurons to connect onto ET neurons. Now, the last thing that I want to discuss are synaptic amplitudes of IT-ET neuron synapses. Going into this analysis, our idea was this. Okay, if a human ET neuron receives more synaptic input connections, then maybe the synaptic weights of each individual connection are smaller to keep an excitatory balance, right? So in our human data set, we found that these IT-ET synapses follow a skewed distribution with a few strong synapses and many weaker ones, and the mean synaptic amplitude amounted to 0.5 millivolts. In terms of short-term dynamics, these synapses display moderate short-term depression. Now, since we know the resting membrane potential and the threshold value of our human ET neurons, we can estimate the presynaptic ensemble size that would be necessary to drive an action potential in these human ET neurons, right? Given that we know the amplitude distribution of the synapses. And when we do this, we come up with the number of 50 IT neurons, approximately 50 IT neurons, that are necessary to drive a human ET neuron. So what about rodents? I want to note that we are currently working on our own rodent data set, but since this is still ongoing work, I will for now show already published data as reference. So this is a study by Kiritani et al., who studied IT-ET connections in mouse M1. And to our surprise, what we discovered is that synaptic amplitudes follow very similar distributions across species. So the human IT-ET synapses are not necessarily smaller. If we take this data at face value, then we estimate that the presynaptic ensemble size that is necessary to drive a mouse ET neuron is very similar with 75 neurons. So this brings me to my summary slide. What our data suggests is that human neocortical layer 5 scaled in such a way that human ET neurons receive more synaptic input connections. Yet, the presynaptic ensemble size that is necessary to drive an action potential in an ET neuron is very similar across species. And therefore, we argue that human ET neurons are capable of integrating more complex information within one cell, and this is why we call these cells giant integrators. Now, of course, more work will be needed, in particular in vivo recordings in human subjects, to determine the exact role of these giant integrator cells. But from the way that they are integrated into the network, we predict that they are likely important for some of the advanced cognitive abilities that we observe in humans. Okay, with this, I want to end my talk. I want to give credit to Sabine Grosser, who's also in the audience. She really contributed a lot to this project. She's responsible for all the anatomical work. And thank you to Professor Geiger for making this possible, and the rest of the lab. Thank you very much.
I wonder if you've considered how this might vary from cortical area to cortical area, because the relative proportion of ET neurons can vary by 50-fold from the front to the back.
Yes.
So it must, and whereas the IT stay quite consistent across. So it must be really different wiring rules in different parts of the cortex.
Yes, this is a very cool question, thank you. So we are focusing on the temporal cortex for this study, because this is one of the regions where this shift in cell types proportion is most pronounced, right? So there's the Joerstadt paper, where the Allen Institute demonstrated that this change in cell type proportions is not the same for all brain regions. But we also recorded from frontal cortex. This will not be in the same study, but we're interested in that exact question. And interestingly enough, the proportion of cells that are classified as ET neurons are also in our data set way higher in frontal cortex. The connectivity from IT onto ET neurons is still quite high, but I mean, we would need more data to have something conclusive there. But yeah, that's a good question, thank you.
Thanks very much. I'm trying to come up with a sensible question, but I don't think I can phrase it, so maybe you can help me out. You framed the original idea as this kind of changing the balance in human brain between cortex and subcortical structures. And I wonder, given the projection nature of these populations, what you think this kind of change in computation might mean for the way cortex interacts with subcortical regions and vice versa?
Yeah, so let me just go back to this slide real quick because I think it's good to have the visualization here. Slow. Okay, maybe not. Anyway, so, I mean, I think it's quite interesting to find that in the human brain, the IT population is drastically expanded, right? This means that different cortical areas are more interlinked, or that there's a stronger connectivity going on within the cortex. Nevertheless, there has to be a copy of the things that are going on in the cortex that is sent to these subcerebral structures. And I think it is very interesting that we find that the human ET neurons receive all the information of, basically this is what we find, all the information of what is going on in the IT network. So they get a very exact view of what the cortex is doing and send this as an efferent copy to the subcerebral structures. Now it is very difficult to speculate what the subcerebral structures then in turn do, but I imagine that there is probably loops, especially if you think about the thalamus, that then goes back to the cortex. And I think that since in humans we have these ET neurons that are giant integrators that receive all this information and can integrate this, that their role will be quite important also for cognitive tasks. But I imagine that there has to be a loop that somehow then comes back to the cortex. Yeah.
Really nice talk. So, of course, these aren't the only cell types that seem to change their proportions even within layer 5. So I'm thinking, like Kostas pointed out, that inhibitory neurons are much more relatively prominent in human. Have you considered how synaptic scaling rules might work for inhibition, like especially in the context, like there's quite a bit known in mouse about how cell type specific inhibition works for this circuit?
Yes, so this is also a very important point, right? So far we are ignoring interneurons in this view of the layer 5 microcircuit, and this is definitely something that we want to do, to include interneurons. Actually, the first time that we saw that the synaptic amplitudes, the excitatory synaptic amplitudes are still large in human ET neurons, right? We thought, okay, maybe that means that there has to be an inhibitory component that counterbalances this. So basically, in my mind, there are two options. Either the expanded IT network in humans is just like the activity is just sparser. This would be something that we could determine from in vivo recordings. Or if the IT neuron is just as active as the mouse neuron, then we will have to have some interneurons that strongly suppress the activity of these ET neurons because they just receive so much inputs, right? But this is something that has to be future work.